Hello friends, today we will learn about antibodies immunoglobulin. Here we will study about antibodies, properties of antibodies, structure of immunoglobulins, immunoglobulin antigen determinants, immunoglobulin classes, abnormal immunoglobulins like multiple myeloma, heavy chain disease and cryoglobulinemia. Now let's begin with antibodies. Antibodies are substances which are formed in the serum and tissue fluids in response to an antigen and react with that antigen specifically and in some observable manner. Now let's know about properties of antibodies. Chemical nature of antibodies is globulin and they are named as immunoglobulins. Serum globulins could be separated into pseudoglobulins that is water soluble and euglobulins that is water insoluble. Most antibodies are found to be euglobulins. Immunoglobulins constitute about 20 to 25% of the total serum protein. Second point is based on sedimentation studies, most antibodies are sedimented at 7S and some heavier antigen bodies that is 19S globulin that is molecular weight about 9 lakh were designated as M or macroglobulins. Let's know about the third point that is Tiselius and Kabat in 1938 showed that most serum antibodies on electrophoretic mobility belong to gamma globulins, which is used now synonymously with antibody. Later, many antibodies were found to migrate as beta or alpha globulins. Now, the term immunoglobulin was proposed by expert committee of WHO in 1964. The name immunoglobulin for antibody has been accepted internationally. Immunoglobulins are mainly synthesized by plasma cells. Immunoglobulins include, besides antibody globulins, the normal myeloma proteins, the cryoglobulin, and the macroglobulin. Thus, all immunoglobulins may not be antibodies, but all antibodies are immunoglobulins. Now, let's know about the structure of immunoglobulin. Porter, Eldeman, and Nisson of in 1900, he was from 1959 to 64, developed a technique for cleavage of immunoglobulin molecule which has led to detailed structure of immunoglobulin. An antibody molecule consists of two identical heavy and two identical light chains. The heavy chains are longer and light chains are shorter. Both types of chains are polypeptide in nature. The two heavy chains are held together by disulfide bonds. Each light chain is also attached to heavy chain by disulfide bond. The H chains are structurally and antigenically distinct in different classes of immunoglobulins. An antibody molecule consists of two identical heavy and two identical light chains. The heavy H chains are longer and light chains are shorter. Both type of chains are polypeptide in nature. The two heavy chains are held together by disulfide bonds. Each light chain is also attached to heavy chain by disulfide bond. The H chain are structurally and antigenically distinct in different classes of immunoglobulins. Five different classes, namely IgG, Ig capital G, Ig capital M, Ig capital A, IgD, D is capital, and IgE, E is capital, and are designated depending on presence of heavy chain like gamma, mu, alpha, delta, and epsilon respectively. The L chain are similar in all cases of immunoglobulins. They are present in two forms, that is kappa and lambda. Kappa is denoted by K and lambda is denoted by L. Each immunoglobulin has either two kappa or two lambda light chains, but both K and L are never found together in a molecule. The light chains are namely kappa and lambda after the investigators Korngold and Lapari. Kappa and lambda chains are present in a ratio of 2:1 in human sera. Now let's know about effect of enzymes. Papain digestion. First point is papain digestion. Potter and colleague split rabbit in Ig capital G antibody to egg albumin by proteolytic enzyme papain in the presence of cysteine. Papain can digest immunoglobulin molecule into three fragments, one Fc fragment and two identical Fab fragment antigen. Two Fab fragments possess the antigen binding site, but the Fc fragment lacks the ability to bind antigen. As the Fc fragment can be crystallized, it was so named. Each Fab fragment consists of a light chain and a part of H chain. The portion of the heavy chain in Fab fragment is named Fd region. Fc fragment consists of parts of both H chains. It is composed of the carboxy terminal portion of H chains. It determines the biological properties of the immunoglobulin molecules such as complement fixation, placental transfer, skin fixation, and catabolic rate. Now let's know about the second point, that is pepsin digestion. Pepsin cleaves at a different point of immunoglobulin molecule and gives rise to Fc portion and two Fab fragments rise to Fc portion and two Fab fragment held together in position. This Fab fragment is bivalent and can still precipitate with antigen. It is called Fab dash whole 2. Pepsin also degrades the Fc portion into smaller fragments. Now let's know about amino acid sequences. Each light chain contains 210 to 230 amino acids, whereas heavy chain has 420 to 460 amino acids. Molecular weight of light chain is 25,000 and that of heavy chain is 50,000. Both L and H chains consist of two proportion each, a variable V region and a constant C region. In the L chain, the two regions are of equal length, while in H chains the variable region constitutes approximately only a fifth of the chain. Variable region are present at amino terminus, that is NH2, and constant C region at carboxy terminus COOH. Antigen combining site is at its amino terminus, which consists of both the H and L chains. The amino acid sequence in amino terminal half of the chain is highly variable. Variability determines the immunological specificity of the antibody molecule. The infinite range of the antibody specificity of immunoglobulins is due to the variable regions of H and L chain. There are many hypervariable regions present in variable portion of the both H and L chains and are involved with the formation of antigen antibodies, antigen binding sites. The site on the hypervariable regions which make actual contact with the epitope are called complementarity determining regions or CDRs. There are three hypervariable regions in L and 4 in the H chains. Now let's know about immunoglobulin domains. Immunoglobulins are folded to form globular variable and constant domains. There are four domains in each heavy chain, one in variable region, that is VH, and three in constant region, that is CH1, CH2 and CH3. There are one additional fourth domain on heavy chain, that is CH4 in IgM and IgE molecule. Light chain has one domain in variable region, that is VL, and one in constant region, in short CL, in all classes of immunoglobulins. Each domain has a separate function. The variable region domains are responsible for the formation of a specific antigen binding site. The constant region domains mediate the secondary biological functions. The area between CH1 CH2 domain is hinge region where enzyme papain acts. The CH2 region in IgG binds CLQ in the classical complement pathway, and CH3 mediate adherence to the monocyte surface. Now let's know about immunoglobulin antigen determinants. Immunoglobulins are glycoproteins and can act as immunogens when inoculated into a foreign species. Differences in amino acid sequences between different immunoglobulin classes, subclasses and types determine their antigenic specificity. There are three major types of immunoglobulin agents determinants. First is isotypes, allotypes, idiotypes. Isotypes. These determinants are shared by all members of the same species. On the basis of isotypic markers on H chain, different classes of immunoglobulins are differentiated. Various H chain markers are immunoglobulins. Various H chain markers are gamma, mu, alpha, delta and epsilon in immunoglobulin, that is IgG, IgM, IgA and IgD and IgE respectively. The light chains are also distinguished through isotypic markers into kappa and lambda. Now let's know about allotypes. These are individual specific determinants within a species. Allotype markers are also present on the constant region of heavy and light chains. These markers are genetically determined. The allotype markers in humans are GM on gamma heavy chain, AM on alpha heavy chains, and KM, originally designed as INV on kappa light chains. More than 25 GM types, 3 km allotype and 2 AM on IgA have been identified. No allotypic markers have been found for lambda light chains or mu, gamma, epsilon and delta heavy chains. Allotype markers are useful in testing paternity. Now let's know about idiotypes. Idiotype markers are located in hypervariable regions of the immunoglobulin molecule. Idiotypes are specific for each antibody molecule. By immunization with FAB fragments, anti-idiotypic antibodies can be produced. Now let's know about immunoglobulin classes. First is immunoglobulin, characteristic of 5 immunoglobulins present in human sera. First is immunoglobulin G, IgG. IgG is the major serum immunoglobulin, about 80% of the total amount. The normal serum concentration is about 8 to 16 mg per ml. Molecular weight is 1,50,000, 7s, half life is about 23 days, longest among all the immunoglobulins. It is the only immunoglobulin that is transported through placenta and provides natural passive immunity to newborn. Next point, it is distributed equally between the intravascular and extravascular compartments. IgG appears late but persists for longer period. It appears after the initial immune response, which is IgM in nature. It participates in precipitation, complement fixation and neutralization of toxin and viruses. IgG binds to microorganisms and enhances the process of phagocytosis. Extracellular killing of target cells coated with IgG is due to recognition of Fc receptor of IgG by killer cells bearing the appropriate receptors. The catabolism of IgG is unique in that it depends on the serum IgG concentration. When level of IgG is raised, as in myeloma and Kala-Azar, the synthesis of IgG against that particular antigen is catabolized rapidly and may result in deficiency of that particular antibody. Conversely, in hypogammaglobulinemia, IgG antibody given for therapeutic purpose will be catabolized slowly. Next point, passively administered IgG suppresses the homologous antibody synthesis by feedback mechanism. Based on this property, isoimmunization of women is done by administration of anti-RhD IgG during delivery. For subclasses of IgG have been recognized. These can be identified with specific antisera. Differences exist in the Fc fragment of the heavy chain of various subclasses. In human serum, these subclasses are distributed to IgG1, IgG2, IgG3 and IgG4. It is protective against those microorganisms which are active in the blood and tissue. Now let's know about immunoglobulin A, IgA. IgA is the second major serum immunoglobulin. This normal serum concentration is 0.6 to 4.2 mg per ml. Second part, half-life is about 6 to 8 days. IgA occurs in two forms, serum IgA and secretory IgA. Serum IgA is a monomeric 7s molecule, that means molecular weight is 1,60,000, while IgA found on mucosal surface and in the secretions, that is secretory IgA, molecular weight 4 lakh, is a dimer formed by two monomer units joined together by a glycoprotein named the J chain. The J chain joins the two monomer units at their carboxy terminals. Secretory IgA and J chains are produced by the plasma cells situated near the mucosal or glandular epithelium. J chains are also present in other polymeric immunoglobulins such as IgM. Now let's know about secretory IgA. Secretory IgA contains another glycine-rich polypeptide called the secretory piece or secretory component. The S piece is not synthesized by lymphoid cells but by mucosal or glandular epithelial cells. It attaches to IgA molecule at their Fc portion during transport across the cells. The S piece is believed to protect IgA from denaturation by bacterial proteases inside such as the intestinal mucosa, which is rich in bacterial flora. IgA is the principal immunoglobulin present in secretions such as milk, saliva, tear, sweat, nasal fluids, clostridium, and in secretions of respiratory, intestinal, and genital systems. It protects the mucous membranes against microorganisms. It forms antibody paste in mucosa. IgA covers the microorganisms to inhibit their adherence to mucosal surfaces. IgA does not fix complement but can activate alternative complement pathway. Now, another point, IgA is mainly synthesized locally by plasma cells and little is derived from serum. Two subclasses of IgA1 and IgA2 are known. IgA2 is predominant, that is 60% in the secretions, but constitutes a minor part of serum IgA. IgA2 is devoid of interchain disulfide bonds between H and L chains. Immunoglobulin M is a pentamer consists of 5 immunoglobulin subunits and one molecule of J chain which joins the Fc region of the basic subunits. Second point, each H chain of IgM has 4 CH domain rather than 3 as seen in H chain of IgG molecule. It constitutes about 5 to 8% of total serum immunoglobulins. The normal level in serum is 0.5 to 2 milligram per ml. Half-life is about 5 days. It is heavy molecule with a molecular weight 9 lakh to 10 lakh, hence also called the millionaire molecule. 9 lakh to 1 million. IgM is mainly distributed intravascularly, that is 80%. It is the earliest synthesized immunoglobulin by fetus in about 20 weeks of age. It appears early in response to infection before IgG. IgM antibodies are short-lived and disappear earlier than IgG. Hence, its presence in serum indicates recent infection. It cannot cross the placenta. Presence of IgM antibody in serum of newborn indicates congenital infection. Its detection is therefore useful for the diagnosis of congenital syphilis, HIV infection, toxoplasmosis, plasmosis and rubella. The valency of IgM is 10, which is observed on interactions with small haptens only. With larger antigens, the effective valency is 5. It is very effective antibody in agglutination, complement fixation, opsonization, and immune hemolysis. It is more efficient than IgG in these reactions. The greater efficiency is due to the binding of complement to several Fc regions of pentameric IgM simultaneously, thus initiating complement cascade and target cell lysis with a single molecule. IgM provides protection against blood invasion by microorganisms. Its deficiency is often associated with septicemia. The isohemagglutinin antibody to typhoid O antigen, endotoxin, rheumatoid factor and WR antibodies in syphilis belong to IgM. IgM monomeric appears on the surface of unstimulated B lymphocytes and acts as recognition receptors for antigens. Treatment of serum with 2-mercaptoethanol selectively destroys IgM without affecting IgG antibodies. This method is utilized for the differential estimation of IgG and IgM antibodies. The two subclasses IgM1, IgM2 of IgM are now we discussed. These are differentiated by characteristic of H chains, that is mu1, mu2 H chains. Now let's know about immunoglobulin D, IgD. First point, IgD resembles IgG structurally. IgD is present in a concentration of a mg per 100 ml in serum. It is mostly intravascular in distribution. Molecular weight is about 1,80,000 7s monomer. Half-life is about 3 days. Like IgM, it is also present on the surface of unstimulated B lymphocytes in blood and acts as recognition receptors for antigens. Combination of cell membrane bound IgD with the corresponding antigen leads to specific stimulation of these beta lymphocytes, either activation and cloning to form antibody or suppression. Two subclasses IgD1, IgD2 of IgD are known. Now let's know about immunoglobulin G, IgE. IgE is mainly produced in the lining of respiratory and intestinal tracts. Serum consists of only traces. It is mostly distributed extravascularly. It is also referred to as regions. Molecular weight is 1,90,000, that is 8s molecule. Half-life is 2 to 3 days. It resembles IgG in structure. It is heat labile, whereas other immunoglobulins are heat stable. It has affinity for surface of tissue cells, particularly mast cells of the same species. IgE mediated type 1 reaction. This is responsible for asthma, hay fever, eczema and produced reaction. High level of IgE in serum is also seen in children with a high load of intestinal parasitism. It cannot cross the placental barrier or fix the complement. IgE is responsible for anaphylactic type of reaction. It may play some unidentified role in defense against intestinal parasitic infections. Now let's know about the role of different immunoglobulin classes. IgG protects the body fluids. IgA protects the body surface. IgM protects the bloodstream. IgE mediates reagenic hypersensitivity. IgD recognize molecule on the surface of beta lymphocytes, B lymphocytes. Now let's know about abnormal immunoglobulins. Apart from antibodies, other structurally similar proteins may be found in serum in some pathological condition. Let's know about these conditions. First is multiple myeloma, second is heavy chain disease, third is cryoglobulinemia. Let's know about multiple myeloma. It is a plasma cell dyscrasia in which unchecked proliferation of one clone of plasma cells occur, resulting in the excessive production of the particular immunoglobulin synthesized by clone. Such immunoglobulins synthesized from one clone of cells are called monoclonal. Multiple myeloma may involve plasma cells synthesizing any of the 5 classes of immunoglobulins, that is IgG, IgA, IgD, IgM and IgE. About 50 to 60% cases of IgA type myeloma involving IgM producing plasma cells is named as Waldenstrom's macroglobulinemia. Apart from excessive production of respective myeloma proteins, Light chains of immunoglobulins may occur as kappa or lambda chains in urine of 50% of myeloma patients. In any one particular patient, light chain is either kappa or lambda only and never both. These light chains are called Bench-Jones proteins, described by Bench-Jones in 1847. BJ, that is Bench-Jones proteins in urine, have a peculiar property of coagulating when heated to 60 degrees centigrade by redissolves at 80 degrees centigrade. Heavy chain disease. Let's know about heavy chain disease. Abnormal heavy chains are produced in excess. This is due to lymphoid neoplasia. Now let's know about cryoglobulinemia. It is a condition in which there is a formation of precipitate on cooling the serum. The precipitate redissolves on warming. This is due to the presence of cryoglobulins in blood. Cryoglobulinemia is often found in macroglobulinemia, systemic lupus erythematosus, that is in short SLE, and myelomas. Most cytoglobulins consist of either IgG, IgM, or their mixed precipitates. Finally finished. Any questions regarding this antibodies, you can ask me in the comment box. I'll definitely answer the question. And subscribe my channel to know more updates about biotech topics. Thank you.